here we are for our second installment of Exploring Inheritance. So now we're going to talk about the variations that are different than just dominant and recessive. Okay, our first one is incomplete dominance. Incomplete dominance means that neither one of the alleles is completely dominant over the other, so you actually do get a blend in the heterozygote form. Okay, so I know we talked about how we had Mendel and he proved the blending hypothesis. False. Well, he proved it false with dominant and recessiveness, but when the blending hypothesis was in place, they thought that that was the only way that any of the genes act, which Mendel did prove that that wasn't correct. But with the blending hypothesis, we can show that some genes in the right format will act incompletely dominant, or the alleles will act incompletely dominant over each other. I show mine with primes. Okay, you will also see that people will show it with a different letter. So you would have big R for your red, just like I have, but then for your white flower, you would have a completely different uppercase letter for the white flower, so big W. And then for your pink flower, you would have RW, okay? There isn't a right way or the wrong way. I just think that it's easier to remember that if my alleles go together, that they have the same letter, okay? But you will see people that write it this way. I do my other allele with a prime. Okay, that's all that these letters and these primes mean, is that it is an allele that is just different than the other one. So our first cross is going to be between a pink flower and a white flower. Okay, same idea, put one on one side, one on the other. Okay, so all of our crossing and all of these different pieces, every piece is going to be the same with how we fill out the Punnett square, how we look at the Punnett square. It'll just be how the genes are going to be expressed in the phenotype. Okay, so really that's the biggest pieces that we're talking about here is the phenotype, and that would be the blending. So we count up how many we have. So we have 1/2 big R prime, or heterozygote, and 1/2 big R prime, okay, or homozygous recessive, or just homozygous in this case. Then we look at what our colors would be. So we have 1/2 pink and 1/2 white. Okay, not too exciting, but we do see that because we have two different alleles and they act incompletely dominant, that they do show the blend of the two other phenotypic ratio or phenotypes in that heterozygote form. Okay, now let's keep going. Here's a couple more examples. Okay, so now we're going to take a pink and a red. Okay, you should be able to predict from the last one that we just did that this time we should get 1/2 red and 1/2 white. Okay, when I fill in, I do leave my capital letter without the prime first. It's just because that's the way that I like to do it, not because that you have to. Okay, but if we were using two different letters, whatever was the recessive phenotype, you would write that one second. So in the above example, when I use the W, you would write W second. All right, so we're going to fill it out. Okay, this time I'll use my percents. So I have 50% big R prime, 50% big R. Oh, I hope you guys all caught my error. Okay, I had primes where I should not have had primes. I was doing the exact same example from before. Okay, so now that we've got that fixed, filling down, filling across, sorry. So then we would end up with 50% big R prime, big R prime, and 50% big R. Then in my phenotypic ratios, we would see the 50% big R, or 50% red, and 50% pink. Okay, so will we ever have just two phenotypes? No. Okay, you always have the option of having 3 different phenotypes when you're talking about something being or acting incompletely dominant. All right, so that'll come out in this next example. So now we're going to cross 2 pink flowers and see what we get. We do see that this is a way that a lot of our flowers get all of the variety of colors that we have. So carnations being able to display all of those different colors is because the genes will blend themselves. So you can take a white and a really dark purple and you'll get a lavender or a light purple. Okay, and that's because those genes are just acting incompletely dominant. All right, so now we count this up. So again, we filled across and filled down. So we have 1/4 big R, 1/2 big R prime, and 1/4 big R prime, big R prime. Okay. Now, if this was a dominant and recessive, then we would only end up with seeing 2 of our phenotypes. But because we have an incomplete gene, we do see 3. So we have 1/4 red, 1/2 we see as the pink, or have the probability of 1/2 of them being pink, and then 1/4 the probability of seeing white flowers. Okay, so we do end up with 3 different options. So in this case, when things act incompletely dominant, we would never have that 3-to-1 ratio that we do with the dominant and recessive. Okay. All right, our next one is what we call codominant. And blood type is a great example of genes that act codominantly. Codominantly means that, again, both of them are they're dominant, but neither one is going to be dominant over the other. With the incomplete, they both get expressed. With the codominant, they both are shown. Okay, so with human blood type, we have type A and we have type B, but if you have an allele for A and an allele for B, you actually have both being expressed. So you are AB blood type. Okay, lots of leaves of our plants, you can see a light green and a dark green. Those genes are codominant, meaning that they are both being expressed. There are some carnations and flowers that will have stripes on them, so they'll have a white stripe and a red stripe. That's codominant. Roan horses and roan cattle have hairs that are white and hairs that are red, so they almost have this like pink hue to them because their hair type is actually codominant. So with our codominants, we're going to use the same kind of example from before, where we've got flowers that have red, red and white, or white. Okay, we're still going to use the primes to show that this is not a dominant recessive gene and it's got something different. So for these problems, you're going to have to look at that phenotype and see, is it red and white or black and white? Okay, what does it look like to see if it is incomplete or codominant? All right, so then we're going to take a red and white crossed with a white. Okay, so we have our heterozygote form, and then we're going to cross it with our white, which has 2 of the same alleles. Cross. Okay, again, I'm just putting my big R without the prime first. It doesn't really matter. Okay. Alright, so then we filled across, we filled down. Now we'll do our genotypic ratio. This should look very familiar to what we got before, but our phenotypic ratios are going to be different. So we have 1/2 big R prime, 1/2 big R prime, big R prime. In our phenotypic ratio, we have 1/2 red and white. I'm not going to write it all out. And then 1/2 white. Okay? So we do see that we now still see our 2 different phenotypes, but this time we have red and white. So maybe they're stripes, maybe they're spots. Okay, maybe it's the edging of the flower, something. Okay, so then we continue. Now we're going to cross our red and white flower with a red. Okay. You should be able to kind of predict what we're going to get from this, but make sure that we're always showing work, because just in case we transpose something differently onto the answer, I want to be able to go back and give you some points. So again, 1/2 and 1/2, big R, big R, big R, big R prime. Okay, phenotypic ratio, 1/2 red, 1/2 red, and white. Okay, so then what should we be able to predict for our next one? If we're crossing a red and white with a red and white, that we have the probability of getting all 3 types of phenotypes. So we set it up, heterozygote, heterozygote, fill across, fill down, and look at that. We have the probability of getting every single one of the genotypes and every single one of the phenotypes. So we had 1/2 big R prime, 1/2 big R, 1/2 big R prime, big R prime. So then that would be expressed by the phenotypic ratio of 1/2 red and white, 1/4 red, 1/4 white. Okay, so when we do cross the two heterozygotes, we do have the possibility of getting all three phenotypes from that cross. Okay, so again, incomplete, okay, is slightly different than codominant. Incomplete, we have a blend in the heterozygote. Okay, codominant, we actually see the shared expression, so we see both of them being expressed, both alleles. Alright, so then that brings us to the things that I kind of hinted to, was the polygenic inheritance. Polygenic inheritance is the one that we all want to know about ourselves. Is where do we get our height? Where do we get our skin color? Where do we get our eye color? Our hair color? All of this stuff that we see and that it makes us so much us is all going to be polygenic inheritance, which means that it's 2 or more genes that affect one characteristic. Okay, so if you've got 3 genes, you're talking about 2 alleles for the one, 2 alleles for the other, 2 alleles. Okay, so you have 3 genes, but you have 6 alleles. Alright, so every time you add another gene, you've got to take those other 2 alleles. So that's why we see such variation when we're talking about polygenic inheritance, and why it's not as simple as saying you're dominant for blonde or you're recessive for this. Okay, now we do have some traits like red hair. Okay, red hair tends to be more recessive. Gray eyes tend to be more recessive. Okay, the amount of pigment that you show in your skin, okay, more recessive versus less recessive. So if you're very, very light, you have more recessives. If you're very, very dark in color, then you've got more. Okay, more dominance for height, less dominant. Okay, but remember that we're talking about multiple genes, and that's why, like in my family, there's huge outliers. My aunt and uncle my uncle, I think, maybe is pushing 5'6". My aunt is maybe 5'4". My cousin, he is 6'7". Okay, because in our family we have tall genes. Okay, so he just happened to get all of the tall genes from mom, all of the tall alleles from dad, and is the tallest in our family. Okay, so that's why polygenic inheritance is so much fun and so cool, is because it's all of these genes and all of these alleles working together to determine and give us the variation that we have in our population. Alright, so then the next one, and this one's a little bit harder, this is what we call sex linkage. Okay, sex linkage means that the alleles are physically on the 2 sex chromosomes. Okay, so we talked about how we have 23 pairs. Well, that 23rd pair is the sex chromosome. And we don't think about them being more than genes for just sex, but they do. They carry actual genes and characteristics on them. So for example, in Drosophila, which are fruit flies, the color, your eye color gene for Drosophila are actually carried on the sex. Okay? For humans, most of the ones that we talk about all actually happen to be disorders. Hemophilia, which is a blood clotting disorder, color blindness, muscular dystrophy, they are all found on the X recessive, or the X-linked, and they're all X-linked recessive. So these traits, even though they're sex-linked, they still act dominantly and recessively while being on the sex chromosome. Okay, so where this gets a little bit confusing is when we're actually talking about who has it and who doesn't have it. So we also have this idea of a carrier. Okay, a carrier is a person who carries the allele and who can pass it down to her offspring, but because of this dominant other allele, does not display the trait. Okay, now the other catch with all of the ones that we'll kind of talk about in class for humans and being sex-linked is that males, you guys only get one allele. Okay, there's only one option. You're either dominant or you're recessive. Okay, so that one allele is acting all by itself because it's not X. Okay, so there won't be anything here on the Y. It's an X-linked recessive gene. Okay, it can only be on the X chromosome. Okay, so here's our first one, and this one we'll talk about it with hemophilia. So we've got a female carrier crossed with a male who has hemophilia. The most important piece here is that you have to have your actual chromosomes written in. Okay, you have to write XX and then an XY. If you can't remember to do it the very first thing. Start reading the question and you see that it's sex-linked trait, start writing down your XX and your XY, okay? Because if you forget them, you will end up with too many alleles, because really we're down an allele in this case because there's nothing over here, okay? Nothing is carried on the Y. So you're still going to fill across, you're still going to fill down, because we're also going to show that we have the 50% chance of a female and the 50% chance of a male, just like you would if you were just crossing for sex, okay? Because I could do this entire thing without the alleles for hemophilia on there and just show that I've got a 50% chance for a female, 50% chance for the male. So I have to have those things on there, okay? Also, when it comes to our genotypic ratio, it's a lot more writing, because look, this is different than this, and this is different than this, and this is different than this. My phenotypic ratios are also going to be different because I've got female, female, male, male, and they're all different. So they're all going to be in this cross a 1/4 chance. So we have 1/4 chance of a female who is a carrier, meaning that she doesn't have it. We've got a 1/4 chance of a female with it. We've got a 1/4 chance of a male without, and a 1/4 chance of a male with, okay? Then when you're just doing your genotypic ratios, so if I ask you a question of, you have Mary who's a carrier and she's married to Joe who has hemophilia, what's the percent chance? Give me their genotypic and phenotypic ratios. This is how you would do it. You would have to show me, okay, and I'm just going to put F or M, female carrier, 1/4 female with, 1/4 male without, and 1/4 male with. Okay, now I could phrase this question very differently. I could have Mary who's a carrier and Joe who has hemophilia, what's their percent chance of having a child without hemophilia? So if I'm just asking a child, okay, what's their percent chance of a child without hemophilia? 50%, because I don't care if it's a male or a female. Okay, now, so that's where you got to be a little bit careful with how you answer those questions. Let's do another one. So now we're going to cross a female with and a male without. Okay, again, I immediately write my sex chromosomes. Then I add my actual allele that I'm crossing. Okay, I'm going to put my big H first because I'm still going to put my dominant before my recessive. Okay, I'm filling across, I'm filling down. X always comes before your Y. Okay, and this is what I get. So I have a 1/2 chance of an X, H, big H, X little h for female carrier, and a 1/2 chance of a male who has hemophilia. Okay, then I do my phenotypic, which I'm not sure what I was trying to write there, so let's erase. So in this case, what I have is a 1/2 chance of a female carrier, okay, and a 1/2 of a male with hemophilia. Okay, and if you guys know anything about hemophilia, this is where this idea of genetic counseling really comes into play. Hemophilia is a horrible, nasty, nasty disease. Little cuts, you don't clot. Okay, your blood just keeps coming out of whatever wounds you have. Okay, so you can actually bleed to death very easily without immediate medical attention. Okay, so if this family, all right, let's use Mary and Joe again from above. If Mary really had hemophilia and her husband didn't, okay, they could go through some genetic counseling. Now, yes, Mary would have lots and lots of medical help with being hemophiliac and having a child, okay, but that's a whole nother story. But they would have a lot of genetic counseling because in this case, do they want to have a little boy? Probably not, because that little boy has a 100% chance of having hemophilia. There's no way for them to have a boy without hemophilia. But if they have a girl, she's just a carrier. She doesn't have it. Okay, so this is the case where maybe going through and actually picking out the sperm that you use, or going through and actually choosing the sex of your child could be beneficial for this family so that they wouldn't have a child with hemophilia. Okay? So that's where genetic counseling comes into play a whole lot. All right, our next one. So we've already had incomplete, where we have blends in heterozygotes, codominant, where we see them both in the heterozygote, sex-linked, where it's actually on the sex chromosome, okay, and now we have sex influence. Everybody wants to know if I'm going to be bald, if I'm not going to be bald. Well, the reason that we say that it's linked on the female side is because baldness is actually sex influence. It's influenced by our flux of hormones in the body. Okay, so when do males tend to go bald? When they're older and their testosterone has started to decrease. Okay, when do females start to go bald? Well, in our case, when females start to go bald, it's because we have less estrogen and more testosterone. So it's all linked to the number of hormones in the body. Okay, so the allele for baldness is dominant in males, which is why we see it more in males than we do in females, but it also is recessive in females. Okay, so kind of stick with me and walk through this one with me. All right, so here it doesn't really matter if this is the male or the female. Okay, I don't have X's and Y's. It doesn't matter. These are the two alleles that the parent, or the two genes that the parents are bringing in. So I've got one person who is heterozygote and one person who has the dominance. And wow, I'm struggling with my B's right now. I'm going to use the primes again just to show, because they are both kind of dominant and they're both kind of recessive, but I want to show that there is some difference. Alright, so I do my cross just as normal. Alright, my genotypic ratios are going to be the same. The change is going to become over here in the phenotypic. So I have 1/2 big B, 1/2 big B prime. Okay, so then over here I have to write an M and an F for male and female. Okay, do I have any big B primes? No. Okay, so for a female, she is going to be 1/2 not bald, okay, and 1/2 carrier. Okay, she doesn't have it, she's not going to be bald, okay, but she could pass on this trait for balding to her child. Okay, now for the male, if this child happens to be male, what's his percent chance of being bald? 100%. Because it is dominant in males, he only needs one of the big B's to become bald. Okay, so that's where the difference is. Alright, stick with me. Okay, now let's do this again. Now we're going to cross two heterozygotes. Okay, so both mom and dad are heterozygotes. Mom would not be bald, dad would be bald. Okay, so we still filling across, filling down. And we end up with this result. Okay, so we've got 1/4 big B, 1/2 big B prime, 1/4 big B prime, Okay, again in my phenotypic ratio, male, female. Okay, so for my male, I have bald, not bald. Okay, so for my male, I have a 3/4 chance of bald and a 1/4 probability that the child would not be bald. Okay, now for my female, it's a little bit different, right? Not bald, oh, bald. Okay, so she's got a 3/4 chance of not bald and a 1/4 chance of actually having a bald female. And yes, it does happen. Okay, so that's where we actually see that we've got some slight differences is in the males and the females, because it is a sex influence trait. Okay, alright, pedigrees. Okay, pedigrees, we will cover these in class just because it's going to be a little bit easier, but we do have some of the symbols. We do show carriers that are the difference, but a pedigree is just a really big family tree of a specific characteristic or trait. Okay, males are always going to be squares, females are circles, carriers are half. Okay, so they could be half that way, they could be half this way. Okay, they're somehow half filled in. Displaying the trait, they're all filled in. Okay, or they'll have something like that. Okay, now the X can also mean dead. All right, we also have a couple other ones that I'll kind of show you in class. So here's married, you show one line in between. Siblings, it's like a fork. Okay, so they're all connected together like that. A divorce, it's got a line through it. Okay. Also, let's say that you have a male and a female who have a child, okay, but they were never married. That's a dash line. Twins, okay, so our fraternal twins, meaning that they're actually going to look different from each other. They are like this, okay, so you could have a boy and a girl, okay, you can also have a girl and a girl. Identical twins will have a line between them to show that they are different. Okay, same pregnancy, that's where the little lines come in. Alright, so if they had another, let's say another sister, that's what it would look like. So that would show that they're twins, but because there's no line, they're fraternal. Okay, and we'll go over these in class just because it'll make it a little bit more fun. Alright, now specific traits that we see for humans. Okay, sickle cell is one of them, and the reason that we kind of talk about sickle cell is because it's a great example of a codominant trait for humans. Okay, so we have blood type and we also have sickle cell. Sickle cell, if a human has two big A's, they're completely normal. If they're heterozygous, they have abnormal and normal. And if they only have the two recessives, okay, they're actually abnormal and will produce only sickle cell. Okay, and the blood cell itself should look like a big old donut with a divot in the middle. Sickle cells look like this little tiny little thing. They don't hold oxygen very well. Okay, they're also very painful because they've got these sharp points at the ends that actually do poke into veins and arteries and actually can lead to huge clogs. So you should be kind of thinking, well, these people probably aren't going to live very long, so how does this still stay in our population? People who have sickle cell do not catch malaria. Okay, and this disease does primarily affect African Americans, okay, and African Americans in developing countries. So they have their heterozygote for sickle cell. They do not get malaria, so they actually survive to reproduce. So these are the people who are reproducing in malaria, or actually these are probably the people who are reproducing even more, who are reproducing in third world countries where malaria is prevalent. Okay, so that's why we still see it, and we'll do a couple little problems with that one. Huntington's, this is one that we will actually kind of dive into a little bit more when we get into our biotech unit after this unit, because we'll do a gel electrophoresis with Huntington's disease. Huntington's is a dominant allele, so you would also think, why the heck doesn't this just go away? It's a dominant allele, you have the dominant allele, you have the disease, you don't have children. Here's the catch. You actually don't really know, for most cases, that you have it until your late 30s and 40s. What have most people done before they're 30 and 40? They've had their children. Okay, it does lead to loss of muscle control and early death and forgetfulness, and it's a horrible, horrible disease. If you guys are house watchers, this is the girl 13, this is what she had, and we'll learn a little bit more about Huntington's kind of as we go. Blood type, okay? You guys need to write this down in your notes, okay? Blood type, we actually do use the capital I and the A on top. I don't know why, okay? But we do use I, and I think it's because it's a multi-allele trait, meaning that we've got 3 different alleles in our population. You only have 2, but there's 3 in our whole population that you can pull from to get yours. Because of this, we've got a lot more combinations. We've got uppercase or dominant for A on both, dominant and recessive. B, you've got a dominant and a dominant, or a dominant and a recessive. Then getting into that codominant, okay? So you've got an A and a B, both are seen in the phenotype of A. Totally recessive, the O, okay? So we're going to do a couple little problems with this. So again, with blood type, you want to make sure that you're keeping your I's there, because if you don't, what am I doing? If you don't, you're going to kind of mess yourself up because you won't remember which one is recessive and which one is dominant. So in this case, we are crossing a person who is heterozygote for A and a person who is heterozygote for B. So we keep our I's. This time we're going to put the A first because of alphabetical order. You're still filling across, you're still filling down, and we end up with a variety of different answers. So we have 1/4, capital I, capital A, capital I, capital B, 1/4, capital I, A, lowercase I, 1/4, capital B, lowercase I, 1/4, lowercase I, lowercase I. Okay? So then again, we're going to have 4 different phenotypes. So you have a 1/4 chance of having a B type blood in this cross, 1/4 chance of having type A, 1/4 of type B, and 1/4 of type O. Okay? Now, our next one is going to be very different, because this one I'm going to cross a person with type AB blood with a person who has type O. Fill across, fill down. Okay? So in this case, right, up here, those people had probabilities, or at least had a 1/4 in each one of their cases of having a child with the same blood type as them. In this case, okay, these people have A, B, and O. They have a no % chance of having a child that has the same blood as them. Okay, so we have a 1/4 chance of capital I, capital A, lowercase I, and a 1/2 chance of capital I, B, lowercase I. Phenotypic ratio then is 1/2 A and 1/2 B. Okay? And my question for you is, why can't a person, so let's say in this case, why couldn't this person that has type AB give to a person who has type O? Okay, so I want you to kind of think about the coagulation effects of taking in the wrong blood type, and we'll kind of talk about it. We will finish talking about all of these genetic disorders in class, or detecting genetic disorders and genetic testing and these types of things, or the end of this when we get into class on either Tuesday or Friday. Okay, so this is for you to review on and look back at. We will be covering pieces of it on Tuesday and on Friday. Thank you so much.